Hi everyone. We are going to look at protein synthesis a little bit more in depth, and I'm going to walk you through it. But what I first need you to do is I need you to get out Bill, because you're going to need to take some notes. Okay? So pause the video, get out note, Bill, and get ready to take some notes. So we're going to kind of step through this process, and you need to be listening, because everything I say is not going to be put up here in words. You kind of have to listen, okay? So we're going to be talking about protein synthesis, and if you remember from yesterday's activity, protein synthesis happens in two steps. You have transcription and you have translation. Transcription happens first, translation happens second. In a eukaryotic cell, so in a plant cell and an animal cell, the first part happens inside the nucleus. So transcription happens in the nucleus, and what we are doing basically is making a copy of our DNA in the form of an mRNA molecule. Basically, you got to think of this as like your nucleus is where we keep all of the originals. So if you think about DNA being a blueprint for the body, the original blueprints are all kept safe and sound in the nucleus. Well, if you want an electrician, for example, who needs a copy of the blueprints, you don't want to give them your only copy. So you make a copy of the blueprint for them, and they can take that out of the office, and then they can go do their job that they need to do. You can make a copy for the plumbers, you can make a copy for anybody. So the copies are in the form of messenger RNA, and it's called messenger RNA because it is taking that message or that code from the DNA out of the nucleus. All right, so we're zooming into the nucleus here, and that is where the DNA is. And what you have is you have the DNA double-stranded, you have a backbone up here, a backbone down here. The RNA polymerase, remember ASE is an enzyme, so you have an enzyme here that is separating the strands. There are hydrogen bonds that are connecting together, and the RNA polymerase is basically separating them so that we can read one side of the code. So each individual then DNA nucleotide has a match, and the base pairing rules are going to be very similar to DNA. A binds with T, but in RNA you got to remember that there are no T's. So there's A will bind with U instead, and then G and C still bind together. Okay, so they call that a complementary binding situation. Okay, so you got to remember that in mRNA, what we're going to be making here, you have U instead of thymine. So basically we have the RNA polymerase is going down the DNA, it's uncoiling it. Here you have more nucleotides binding, and they're U's. See how a U bound there? And another U, and another U, and another U. That's how you know it's RNA. And so what happens is this RNA polymerase is going to move down the strand of DNA, opening it up, and you're going to read the code that is on the DNA, and they're making an mRNA strand from that. Okay? Now, what will happen at the other end is that the DNA is going to coil back up again. Okay, so it's basically opening so that it can be read, so that the code can be read. The mRNA strand is just using it as a template to match up the nucleotides, and then the mRNA strand is going to be released. Okay, so we're not doing anything to the DNA, we're just using it as a template to make a copy from, and then it'll coil back up. Okay, so one thing to know about the mRNA is that it is single-stranded, and we know it's RNA and not DNA because it has U's. We know this is DNA because there's T's. We know this is the other strand of DNA because there's T's. Only one side of the DNA gets read. All right, so let's see what happens next. Once transcription is complete, so that was transcription, okay, basically reading the DNA, making an mRNA copy. That happens in the nucleus. Once that's all done, the mRNA There's some other monkey business that happens with the mRNA, but we don't have to get into details with that. Basically, the mRNA is done. It can leave the nucleus, okay? So there it is, leaving the nucleus. And what you see here, there's this little blue thing here. Well, that's a ribosome. Ribosomes are another organelles. We learned about those when we were learning about parts of the cell. And the ribosome is going to be important for this next step. So you see it coming, and it's going to bind to the mRNA that just left. So this is translation now, okay? Transcription all happened in the nucleus. Translation now is just out in the cytoplasm. It's happening at a ribosome, and it's basically taking the mRNA code, okay? And each code, or each part of the mRNA, you're gonna look at the strands of the nucleotides. Every 3 nucleotides is a codon, and those codons will code for a very specific amino acid. Remember, amino acids are the building block of the subunits of protein, okay? So these guys are tRNA. The tRNA has what we call an anticodon. So if the codon is mRNA, the anticodon is on tRNA, and it's basically the match, okay, of what is on the mRNA strand. But what's neat about the tRNAs is they're carrying, see these letters? LEU stands for leucine and valine and threonine and cytosine. They are carrying amino acids, okay? And the codes are always going to be very specific to the amino acid. The anticodons are going to match the codons that are on the mRNA. So we see here, this says AUG. Well, what does A bind to? A normally binds to T, but we're talking about an RNA here, so what does A bind to? A will bind to U. U binds to A, so now we're looking for a codon, anticodon that is U, A, and G binds to C. So we're looking for U, A, C. We find one over here that says U, UAC. Oh, it's not gonna let me do it. Hang on. So here is UAC, and that binds. And what happens is that binds, the thing shifted down, ribosome shifted, and now it exposes a new part of the codon. Now we have GUA. G binds with C, so we're looking for one with C. U binds with A, so CA, and A binds with U. So we're looking for CAU. So we gotta find one that says CAU, which is valine, okay? And now, what you may have noticed is that the tRNA that was here, that left, but the amino acid it's carrying stayed, because now it is bound to the valine. The ribosome shifted, and now another tRNA is going to bind. This says CAU. So you gotta find one of C. It's gonna start with a G. G is gonna bind with C. A is gonna bind to U. U is gonna bind to A. So we have histidine. Again, and now we have 3 amino acids in a row. Here is AAA. So we're looking for UUU. A is binding to U. C binds with G. So we're looking for like that guy. G binds with C. So we're looking for one that starts with C. U binds with A. So it's CAA. So we got another valine. And notice that this peptide, that's what we call this, a growing protein, is getting longer and longer and longer. So then U is going to bind with A. So let's see. A, C, G, C, A, C. So that was going to be G, U, G. G, G, U would bind to C, C, ACA, bind to UGU. The rest of these, and then UAA actually codes for what we call a stop codon. So then the stop codon tells the amino acid that we're done building the protein. So it allows the ribosome to be released, and then the chain of amino acids is now released, and those amino acids can then go off and fold up, because if you remember, the proteins are like a folded structure, and they have a function based on the order of their amino acids and based on the shape that they make. Okay, so the final product is a protein, and that's going to go off now and do its job. Okay, so that's pretty much the whole process of protein synthesis. Okay, one more time, quicker. Nucleus transcription is going to happen in the nucleus. RNA polymerase uncoils the DNA. mRNA nucleotides read it and bind, use the DNA as a template to read the code. And because it is making a complementary strand of mRNA, this message is being created. It's a copy of the message, essentially. It's going to go ahead and leave the nucleus. It's going to bind to a ribosome. Once it's at the ribosome, basically the ribosome is acting as a place, a location. The ribosome doesn't actually do anything other than provide a space to reveal the codon. Okay, so that gets exposed. You're going to start bringing those in as the code continues, and then it'll be released to be a protein. Okay, so hopefully that helps paint a picture of protein synthesis.